Good morning, students. Our today's topic of discussion is chromosome. Before we go into details about what a chromosome looks like, what a chromosome does, let us find out what is a chromosome. A chromosome is a highly coiled and condensed chromatin fiber. So now the question arises that what is a chromatin fiber? Chromatin fiber is nothing but a long DNA molecule that is packaged or that is associated with many proteins. So let me quickly say all of this again, that chromosome is a highly coiled and condensed chromatin fiber. Chromatin fiber is in turn a fiber of long DNA molecule with associated proteins. We know that chromosome is present in most of the cell's nucleus and is responsible for genetic information that we carry. Scientists have observed chromosome under electron microscope, and there they have found it to be obviously they have to stain it to observe it under electron microscope. They have find it to be resembling fibers and looking like the alphabet X, of English language. So let me share my screen and show you what a chromosome looks like under electron microscope. So this is what a chromosome looks like under electron microscope. Here you can see that these are deeply stained and pinkish stain they have taken, and yes, they resemble a X-like structure, isn't it? And it can be observed in great detail. Whenever a cell prepares to divide, at that time the chromatin fibers, they actually coil and they condense further and becoming pretty thick that they can be distinguished as chromosomes. So these chromosomes at that stage, they pick up certain dyes and get colored. So that's why we can see them, and that's why they are named like that, because the word somes, it comes from the word soma, which means body, and the chromo part of the word chromosomes comes from the word chroma, which means colored. So during the division of the cell, when the cell starts preparing itself for division, at that stage, these chromosomes can be distinguished in this form, because otherwise it is present in the form of chromatin fiber inside the cell nucleus. But this extreme condensation takes place just before the cell prepares itself to divide, and the chromatin fibers get further condensed. So now we'll come to know about what chromatin fibers are made of. As I already told you that chromatin fibers are made of long molecules of DNA associated with proteins. So basically we can say that chromatin fibers are constituted of two parts. One is DNA molecule. It consists about 40% of the chromatin fiber, and the other part is, the protein part is histone proteins, which constitute about 60% of the entire chromatin fiber. Now, this DNA molecule, its shape, it was supposed to be studied by scientist Dr. Rosalind Franklin, but later on Dr. Watson and Dr. Crick, they came up with the structure of DNA, which is also known as the double helical structure, and they were awarded even the Nobel Prize for that. So let us see what this special DNA structure looks like. Okay, so I'm going to present the screen again. So we'll see what this structure looks like. So it's the same chromosome that we have seen. And here comes the DNA molecule. So you can see that there are two strands here. One is a pair of parallel strands, another is a coiled strand. Actually, the structure of DNA is like this double helical structure, the coiled one. Parallelly, it is shown to explain it to you like nicely. Now, a single DNA molecule is huge. It's large, very large, and hence it is often called a macromolecule. It is made up of two strands, and the two strands are complementary. And let me repeat again, they are not parallelly arranged. They are arranged, they are wound around each other in a double helix. Now let us notice the parallelly arranged strands. Each DNA strand, let us notice that one strand. Each DNA strand is composed of repeating nucleotides. Now, what are these nucleotides? They are basically made of three components: phosphate, pentose sugar, and a nitrogenous base. Now you can see that it is labeled sugar phosphate backbone. Now, the sugar phosphate backbone contains the nitrogenous base, which is attached to the sugar, and it is inwards. The nitrogenous base is inwards. And this nitrogenous base is joined by hydrogen bond to its complementary nitrogenous base on the other strand. So thus you can see that the two strands, they make they look like a ladder where each nitrogenous base looks like the rungs of the ladder, isn't it? Now, I told you just now that each nitrogenous base joins with its complementary nitrogenous base. So let us know what nitrogenous bases are there and to which nitrogenous base each nitrogenous base gets joined to. In every strand, in a DNA strand, you will find there are 4 types of nitrogenous bases. They are represented as A, G, C, T. A is for adenine, G is for guanine, C is for cytosine, and T is for thymine. You can see here that A is linked only to T and G is linked only to C. Nowhere you will find that A is linked to G or A is linked to C, or other than this combination, any other combination you will never see. So adenine will always join with or pair with thymine with the help of 2 hydrogen bonds, and guanine will always pair with cytosine with the help of 3 nitrogen bonds. Okay, so this is what a typical DNA molecule looks like. So let us come back. Yeah, so now that's the DNA part of the chromatin fiber. Let us come to histone. It is a protein. And how is histone arranged in the chromatin fiber? There are 8 histone proteins, or we can call it histone octamer. They are arranged as a core, and around it, the DNA strand are bound. DNA strands bind the 8 octamer, or we can say 8 histones, histone octamer. So you can imagine it as a football bound with a rope in 1 or 2 loops. Okay, so a single human chromosome And this structure of 8 histone proteins at the core, bound by DNA molecules, a long strand of DNA, is called as nucleosome. And each human chromosome consists of millions of nucleosomes. Okay, so let us break down the entire structure and understand this. DNA molecule and histone proteins are there. 8 histone proteins come together, forming a core of histone octamer, around which the DNA strand is wrapped. One such unit is known as nucleosome. There are many such units. They are getting condensed, they are getting coiled further, forming a fiber. Those fibers are known as chromatin fibers. That's how the fibers are present in our cells. Just before the division, when the cell prepares itself for division, the chromatin fibers, they start getting condensed and coiled even further, forming the first picture that I showed you, the chromosome, forming structures that look like the chromosome. Let me show you this entire scheme in the form of a picture. I have to share the screen one more time. Here it is. Yeah. So see this. I went past the diagram. Yeah. See this here. We can see this double helical DNA molecule. They are getting coiled, and here you can see 4 purplish histone protein, and at the bottom of that again you will have 4 histone proteins forming a histone octamer. And see, the DNA strand is wrapped around this histone protein, it's bound by another histone H1, and see, so many such nucleosomes are present. Each such unit is known as a nucleosome. There are so many such nucleosomes are present which bind and get condensed even further to form fibers. These fibers are called chromatin fibers, which get condensed even further, forming chromosomes. It's like a coil getting further and further condensed. Now, this chromosomes that we are talking about, let me tell you one interesting fact, that each human cell, they contain approximately 2 meters of DNA if they are stretched end to end. And you know that how tiny a cell is, and a nucleus is tinier than that, and all that is fitted, the 2-meter long DNA is fitted into the nucleus, which is only about 6 micrometer in diameter. So you can assume how tightly it has to be coiled. It has to be super coiled. Now, let me come to the first picture that I showed you. In this picture, I told you that it looks like X, alphabet X. Now, it looks like alphabet X because each of those, when they are visible, when the chromosome is visible in its condensed form, each chromosome consists of 2 sister chromatins. The 2 arms of X, they are actually 2 sister chromatins. And if you notice the entire length of the chromosome, you will see that at one particular point, there is a constriction. It doesn't need to be at the same point for every chromosome. Every chromosome has a specific point. This constriction is the place where the sister chromatids are joined. And this place is called centromere. Now, this centromere is very important part of the chromosome because this is the place where spindle fibers, another type of fiber, spindle fibers, get attached during cell division. And it is the spindle fibers that help in the separation of the sister chromatids during cell division. That is a topic for another day, but let us now quickly recapitulate today's learning. We learned about what DNA molecule looks like, how it is arranged with a histone protein. They make chromatin fibers, and that gets supercoiled to form a chromosome. So this is what is going on inside you, inside me, inside each of myself. In the next video or in our next class, we'll talk about cell division and what role what role chromosome has to play in cell division. So thank you for today. Have a good day.